All right, we're recording. All right. Good morning, everyone, and welcome to day 3 of our protein crystallography summer collaboration. Just a reminder for students to continue to work on your Google Slides. You could start adding them to the existing slide that has student and teacher introductions in it. That's the very first slide of this week, which you'll find in the Google Classroom. You'll find that in the stream with the classwork. So just make sure you are working on that and get that done, so this way we can get to meet everybody and get to know who everybody is. I'm going to switch my tab over. Today we're going to continue discussing about the relationship between X-rays, freezing, and proteins, and that's a big thing. The X-rays play a major role in how we are actually able to see what's going on with our proteins, and in order to use those X-rays, we have to do what Mr. Pinto talked about yesterday, which is freezing the proteins. And in order to freeze the proteins, you have to make sure that your proteins can actually work, and that happens. I don't know why this went to the end. I must have been on this. And that only works well when we are able to correctly produce the crystals, which is what Mrs. Mackey had spoken about. So the image, can everybody see my screen? I'm on, right?
Okay. Yes, you are. You're on. You're good. You're good.
Right. So this little image here shows at the bottom here, let's see if I can get my little pointer thing going here. Yeah, this works. So at the bottom here, I don't know if you can see it or not with a pointer. There is a structure of a protein in there. So there's a crystal in there, and the X-rays hit that crystal, and then those X-rays, when they pass through that crystal, they are diffracted onto this detector, and there's a dot pattern that forms, and that allows a computer to take that dot pattern and transform it into a three-dimensional structure. So the real key thing here is getting the X-rays. Now we've all, most of us have had an X-ray of one form or another, a broken bone or, something that was fractured. And we know that X-rays can pass through certain tissues, and then they can create an image based upon what we want to see. So most of the time we can see a bone, but we also can use the X-rays to actually see where the individual atoms are located within a particular compound. And so we can use this for proteins, we can use it for DNA, we can use it for a lot of different things. And at BNL, we don't just have X-rays being used to look at protein crystals. X-rays can be used to look at a whole wide range of different types of materials, both organic and inorganic. It can be used for chemical analysis, it can be used for looking at electronics and all sorts of incredible things. So you need to generate X-rays, and in order to do that, you need to use something that can make those X-rays in a controlled environment that can then focus the X-rays to be as precise as we possibly need them to be. And that happens at a thing called the National Synchrotron Light Source. So we need X-rays that have high energy, so you can actually see the individual atoms, and they have to be focused too. You can't have X-rays, zapping out all over the place. So the tool that does this is the Synchrotron Light Source 2, which is one of the brightest lights in the world. And as Sharon had mentioned in her introduction, the center of that ring, which is that open field in the middle, Yankee Stadium can fit inside of that. So that's how large this is. And it needs to be that large so that we can generate X-rays at a high enough energy so that we can see these very precise, tiny differences in the placement of individual atoms within the molecules. Okay, so this is an overhead view of the National Synchrotron Light Source. There are 5 buildings that are associated with it, which look like these little rectangles on the outside of it. And those are known as lab operation buildings, and that's where there are wet labs, and that's where the beam lines are located. And then this ring here is where we see the beam lines, and there's a ring in there that contains the electrons that are spinning at a very high rate of speed, so high that they're just about close to the speed of light. And those electrons will emit X-rays, which are then captured by the individual beam lines. This is what it looks like if you lift it off the roof. You would actually see this ring, which is called a storage ring. And then these tiny little lines that come off, those are spokes that represent the individual beam lines. And our beam lines are located right over here in building 745. And there are several beam lines that are used here exclusively for X-ray crystallography and X-ray diffraction. This light source starts off with a linear accelerator and a booster ring that gets the electrons up to the speed they're supposed to be. And the way that they move the electrons is actually by physics. It's using magnets. And so you can see up close, that's where our X-ray diffraction beam lines are. And the way that this works is electrons are first shot into this booster ring through this linear accelerator. And the linear accelerator captures electrons that are fired off by an electron gun, which heats tungsten to about 1600 degrees. The electrons move in this small ring and are gradually brought up to almost the speed of light. And once they're brought up to that speed, they're injected into the main storage ring, which is indicated by number 4. And that main storage ring, the electrons just circle through that ring. And as they go through that ring, there are large magnets that slow them down just a little bit. And as those electrons slow down, electrons get emitted. X-rays get emitted. And those X-rays are then captured by these individual beam lines, which are represented by these little boxes with these arrows. And it's at the end of these beam lines where we have these stations that allow the atoms in proteins to be seen by taking the X-rays and focusing them and filtering out different wavelengths so you get precisely the wavelength that you want to hit your tiny protein crystal exactly where it needs to be hit. And all this needs to be done maintaining a high energy level so we can actually see the individual atoms. Okay, so this is just a quick look at some of the magnets and the other devices that affect the electrons. So this is the storage ring, and there's a tube here that the electrons are moving through. And these magnets, there are different types of magnets. Some of them help focus the beam and keep it centered. Others bend and slow the beam down so that as the electrons pass through that blue set of magnets, they slow down and they emit X-rays, which then move down these black pipes. And those pipes eventually feed into the beam lines where we can then have specific data collection going on. Then there are these other devices that help undulate. They help to kind of bring the electrons to the center of the beam where they're supposed to be. They also help to maintain their velocity because they need to be at 99.9 something percent the speed of light. So you got to keep these things moving fast. Those magnets are electromagnets, so they generate their magnetism by having electricity pass through them. So this beam line, actually all the beam lines generate a tremendous need to have a lot of electricity in order to maintain the magnetic effect of keeping the beams going and of keeping the electrons moving. So there's a tremendous amount of electricity that is used by this facility, and this facility, when it's running, runs 24 hours a day, 7 days a week. So you can just imagine the amount of electrical energy going into this to make this work. This is a close-up view of the actual magnets that are inside the storage ring of the synchrotron light source 2. There are different types of magnets. There are magnets that are organized in groups of 6, 4, and 2. And there's a silver-looking pipe here. That's the actual pipe that the electrons flow through. And you can see all these wires here that are hooked up that basically power these magnets. These are very powerful electromagnets that require the electricity to run them. And you can see how these alternate. You got a group of magnets here, then further on in the distance, you see there's blue, then there's this red, orange, yellow, then there's blue, red, orange, yellow. So they have all these magnets alternating with each other to help maintain the velocity and the focus of the different electrons as they're coming through. This is another close-up view before these magnets are being put into position, and you can see that they're not quite hooked up yet. But on the top here, you can see these little forks. These look like plugs, and that's actually what they are. And they get plugged in to the electrical system, and that's what runs those magnets. The magnets also generate a lot of heat, and so you can see that there are these 2 supply lines here. This is for liquid nitrogen, so they need nitrogen to keep these cool so they don't overheat. And then the round circle here off to the left, that's the beam line itself. Not the beam, that's the actual tube that the electrons flow through. Okay, and the white things here are the magnets, so that's a dipole. The yellow one here, I think, is a quadrupole, and then the red one and the orange one there. The orange one has 6 magnets in it. So There's a lot that goes into making this happen. They sit on these giant steel girders that keep them stabilized with shock absorbers on them because we can't have them vibrate. You can actually detect the vibration from the crashing of the waves on the South Shore on Smiths Point and Robert Moses and Cupsogue. That can actually be detected in the floor of the facility. And so they have it sitting on these dampers and shock absorbers that minimizes any movement they have. And you guys remember when we had that earthquake earlier in the year? The whole light source shut down because they detected a change in the positioning of the beam because of the vibration from the earthquake. And so they had to shut down for a few hours to make sure that everything was all situated. And it automatically did that too. So it automatically detects if there's a change in the trajectory of the beam. This is one of the beam lines. So these are some of our students from Spark from a few years ago. And the beam lines basically are set up with a lot of computers that are connected to the detectors. And the detectors will give you an image of what your diffraction pattern looks like, which is that orange screen that you see. And then above the two people's heads to the left, you can see that that's where the crystals are located inside of the loops that we use to collect them. And then off to the left side is an image of what the protein looks like after the X-rays have hit it. So at the beam line, we can analyze a diffraction pattern. We can have that diffraction pattern refined very quickly and get a basic rudimentary look at what the protein looks like. So there's quite a bit going on at the beam lines. And again, just to recap what Mr. Pinto talked about yesterday, we freeze the crystals in liquid nitrogen, right? And those crystals are then placed inside of the puck. So that's an actual puck that one of our students is working on. And those white little dots indicate the small little pins that contain the cryo loop that has the protein in it. And by putting it in the liquid nitrogen, it preserves the protein and it maintains its stability. So this is what the spine cap looks like, and that's the little loop at the top of the cap that's used for harvesting the crystals. And if we zoom into that loop, that's what one of the protein crystals actually looks like in that loop. So this is a picture that I actually took from the beam line while we were preparing the crystal for data analysis. So soon after we took this picture, we shot it with X-rays, and we were able to get a diffraction pattern of what that protein looks like. This is lysozyme. These are some lysozyme crystals that we captured. This was done actually under a plain old light microscope in my classroom. So I was just able to angle the camera the right way so that you could actually see how the proteins are organized inside of that loop. And it's in that loop that we want to shoot it with the X-rays so that we can get the exact structure of the protein. And so here's another picture of same thing under a light microscope of a lysozyme crystal. And the idea is that we get those crystals in that loop mounted on the structure called a goniometer, right? So the picture to the left, there's a tiny little magnet on that, and the pin with our loop on it will actually be attached to that magnet. And there's a robot that mounts it, and the robot here is a green one. We call it Kermit. Like Kermit the Frog. That's actually what the scientists named it that, not us. It'll mount the crystal, and then that will allow the X-rays to pass right through it. So the X-rays will actually come from the top of the screen, pass through our crystal, and then there's a detector that's off screen that's at the bottom, and that's what will pick up the diffraction pattern once the X-rays hit it. This is Big Bird, and at the end of Big Bird, you can see it has a little googly eye here. There's this long, kind of frosty looking structure. That's called the gripper. That will actually pick up the individual pins from the pucks. And the pucks are stored in this giant liquid nitrogen dewar. And the dewar is just a big storage container. It's like a giant freezer that keeps the pucks in there. And so the end of this gripper will grab one of the pins, and then it will precisely place it into the beam so that we can then analyze the protein structure. Okay, so that's one of our scientists, Dr. Soares, showing one of our students how the robot works. Everything is robotic inside of the beam line. This way, scientists don't actually have to be in there. And this is what the pin looks like when it's mounted to the goniometer. And you can see right at the end here, there's a tiny little white speck. That's the protein crystal frozen. And the X-ray beams will come from the top and move through that crystal and diffract and create a diffraction pattern. The other thing that we also have is we have cameras mounted there, and we have a thing called a cryostream, which blows liquid nitrogen onto the crystal to keep it frozen, because it's in an open air environment and basically it's room temperature in there, and we don't want the crystal to melt. We don't want the protein to change shape. So we keep a cryostream on there so that it's able to maintain its frozen nature. And that's what that crystal looks like on camera. And so what we'll do is we can use the computers to precisely position the crystal so that the X-rays can hit it. When the X-rays hit that crystal, it produces hopefully we get a beautiful diffraction pattern. And before it does that, what the computer does is it tells you where it seems to have picked up good diffraction. So you'll actually get a grid that the computer will generate over the crystal, and it highlights hotspots where, based on the diffraction that came through, it's where the computer has detected that there is some sort of diffraction. And then it's up to us as the scientists to determine whether or not that diffraction is actually good diffraction. Do we have a good pattern, or is it just ice, or is it just, background noise? So the computers really do help us a lot, and they give us some suggestions as to where we can find potentially really good diffraction data. And so these are some really small crystals that are in one of the loops, and the little plus sign here indicates the precise point where the beam is going to hit. So what we do is we align the crystals so that they are right underneath one of those, that plus sign, so we know that we're precisely hitting the crystal the way we need to. And then we do that. You can zoom in real close here. So this is a real close zoom in, and obviously this is not positioned right because the plus sign is up here. So we would not even be zapping any crystals, but this would get moved so that that plus sign is over one of these beautiful crystals. And then we can collect data on it. And when we collect data, you can see this is a giant crystal. This is actually from earlier this year. Off to the right-hand side, you see all these tiny dots here. Those represent the diffraction pattern from that particular crystal. And so that particular crystal gave us a really nice diffraction pattern. So this is what it looks like. The X-rays hit the crystal diffracts, we get these dots on this film. Now, the film is actually on a detector, a computer detector, that will then take those dots and transform it into a 3D structure for us, okay? What we want to see is a nice diffraction pattern with nice concentric dots. We want to see that there's a pattern that forms, there's uniformity there. And then what will happen is the computer itself, there are programs at the beamline that analyze the pattern, and then they use that pattern to create a structural model. So from this pattern of little tiny dots, we have computer programs that can convert that into a three-dimensional structure of what a protein should look like. So this is just a real close-up view of decent diffraction. It's not super great, and you can see the difference here that these dots don't really have a lot of pattern to them. They're kind of scattered randomly, where in the previous slide you can see that there are these streaks and these lines, and they look like they're all in a row. That's kind of what you want to see. You want to see some sort of organized pattern to the dots, not something random. Like this looks like the night sky when you're looking up and you just see like a couple of dots scattered all around. You want to see some uniformity there, okay? And when you do that, you're able to convert that pattern into a structure of a protein. So what this picture shows you is these blue clouds. That represents the electron cloud of where the electrons are located for the atoms that form the protein. This cloud was generated by using this diffraction pattern, okay? So from that diffraction pattern, you can get this cloud of where the protein is, and then we use a protein that's similar to it, like an analog, and we overlay it onto this cloud, and then we try to create a structure based upon the cloud. So the cloud is our actual data that we collected, and that's the actual structure of our protein. But to build it from scratch would take way too long. So what we do is we take a protein that's similar to the one that we're interested in, and we overlay it on there. And then we try to fit that protein into that cloud. And once we do that, that becomes the actual structure of the protein. It's known as molecular replacement, because what we're doing is we're replacing the known model with amino acids that we know fit the existing data, okay? And that becomes our new protein. And so we do that with a computer program that's known as COOT, and that is known as manual refinement. And so students and teachers, you will all learn how to do this, where you'll actually manually adjust this yellow backbone of the protein to fit these clouds so that we have an actual structure of the protein based on that. And then the little X's here that are red, those represent water molecules. So these little round spheres are basically where there's a lot of water. And proteins are surrounded by water. They exist in a watery environment. And the solutions that we mix them in, they have water in them. And so it'll freeze up, and so we'll see that. And so we can determine what's water and what's not based upon what we have in there, okay? So why X-rays? Okay, why is it that we want to use X-rays? What X-rays allow us to do is get better resolution of the bonds and the electron densities of individual atoms. So if we didn't use X-rays, we wouldn't be able to see the actual individual placement of the atoms and the bonds in the molecules. So this is really like a microscope that's being used for organic molecules. So crystallography allows us to get that three-dimensional position of the atoms. So it's not just getting a flat picture of it, but it's actually getting a 3D picture of it because the X-rays can go through the molecule. So basically we go from that single crystal, which was frozen, to a diffraction pattern, and then that diffraction pattern is converted into an electron density map, okay, which is that blue cloud that I showed you on the last slide. That map, when we position the atoms correctly in that map, that allows us to then generate a protein model. And once we have a model, we can then deposit that to the protein data bank, and that model can be used to help inform scientists of the structure and the unique aspects of each protein. Okay, and that's the reason why we're using those X-rays. So what's actually happening at the molecular level and physically is as the X-rays pass through the crystal, they actually make an interference pattern. But what happens is there's a repeating pattern of atoms within the crystal. If you remember yesterday, the crystals contain individual molecules that are lined up with each other, okay? And if they're oriented correctly with each other, that interference pattern is reflective of the structure of the actual sample. And we can actually look at the distances between the different planes of the atoms and determine how they're arranged within that particular lattice of individual protein molecules within the crystal, okay? And that allows us to get the pattern that we want. So you need to use X-rays because X-rays are small enough to basically decipher the difference between different types of atoms. So we can tell whether it's a hydrogen atom or a nitrogen atom or a carbon atom or an oxygen atom, okay? So we can determine that, and the computer can pick that up and tell us, okay, this is an amino acid, and here's where the different carbons, hydrogens, nitrogens, and oxygens are located within that particular amino acid. So it's pretty cool that we can actually drill down to the amino acid level and look at where the actual individual atoms that make one amino acid are located. And then that helps us determine the actual structure of the protein, okay? And again, the densities and the diffraction patterns generate that model for us, right? And this is what's going to take us the most time. Collecting the data actually doesn't take that long. It's a few hours during a day at the lab. Analyzing the data can take weeks, if not months, because you have to go through this and painstakingly position it so that it fits the cloud data. So if you look at this picture on your screen, you can see that these green areas, they're all sticking out all over the place. They're not really covered by this cloud. So that doesn't fit really well. So we have to go and play with this a little bit and use the computer program to try to see how we can get all of these little branched areas to fit in that blue cloud. And sometimes it doesn't work, and that means that you didn't get good data. So you have to go to another data set, and you have to look at another crystal. Or sometimes you have to go back to the drawing board and shoot the crystal all over again, okay? I want to play a real short video on X-ray crystallography so you can kind of see how this all works in action. This was actually a video that was taken from another beamline that's over in, it's the Diamond beamline, I believe. It's over in Great Britain.
And as they wiggle, they produce an intense pulse of electromagnetic radiation in the form of X-rays. and that pulse of X-rays shoots straight into one of the lead-lined experimental hatches where visiting scientists carry out their diffraction experiments. During the experiment, the crystal, which is mounted on the end of this little pin, is cooled by ultra-cold nitrogen gas from this nozzle to help it withstand the punishing radiation. This allows us to take much longer exposures, which are needed when working with very small crystals. The X-rays from behind emerge here and are scattered by the crystal into many different angles. The diffracted rays are captured on this detector, which can record up to 25 images a second, much faster than the photographic plates that we used to use.
So what do we do with this information? How do we convert these diffraction patterns into molecular structures? Well, I can see my crystals down a microscope like this fairly easily. What I'm doing here is using lenses to make a magnified image of a tiny object. And that works because the lens can recombine or focus the light scattered from an object to make an enlarged image. Because a molecule is much smaller than the wavelength of light, I'm never going to be able to see it using an instrument like this. Instead, we use X-rays, a form of light that has a wavelength about the size of an atom. But there are no lenses to focus X-rays, so we use our mathematical understanding of how light scatters to do the work of the lens for us. And that allows us to work backwards from the diffraction pattern to a three-dimensional image of the protein in the crystal. What we end up with are three-dimensional maps which plot the electron density of the protein molecule, and this reveals its branched, chain-like structure. The resolution of these maps is dependent on the quality of your crystal. The better they are, the more accurate your model is likely to be. Originally, crystallographers would painstakingly build these 3D models by hand, but now we rely on computers to generate them. With good quality data, these maps are stunningly detailed. You can see the lumps and bumps of every atom, every twist and turn of the protein chain.
Even after you've spent your time at Diamond and solved your protein structure, you've really only taken the first step on a much longer journey towards understanding how it works. But from these sorts of investigations, we can learn how the biological molecules of your body interact with one another, or how to design drugs that will bind to them more effectively, or even to design proteins that nature never got around to inventing. Even after 100 years, these are still exciting times to be working in structural biology, thanks in no small part to facilities like Diamond.
And also thanks in no small part to facilities like the National Synchrotron Light Source as well, right? Right, so just to kind of wrap up, these are the structures that we've resolved using X-ray diffraction. So these were the first two structures that our collaboration resolved several years ago. 6UEV is the code number, and 6PEY. So these were crystals that were generated by students. They were harvested by students. The data was collected by students, and the structures that you see here on the screen were resolved by students. And so these are now in the protein data bank. And these three here are the most recent ones. 8TXA, the one on the left, was actually deposited a year ago in August. And 8FCH and 8FCI were deposited earlier in 2022. And so these have all, again, been structures of proteins. These are structures that were resolved by students, and they were crystallized by students and collected by students. So this is the type of thing that you'll be doing moving forward at the Beamline. And again, what's important is to understand that the crystals need to be in as pristine a condition as possible. We want the most ideal structure to the crystals so that when we shoot them with the X-rays, we can get the most precise structure out of them. The better the crystal, the better the structure we're going to get. And that's what was mentioned in the little video that we had there. Okay. So do any teachers want to comment, or do we have any questions before we wrap things up?
I just had a quick question.
Go ahead.
In the crystal, you have multiple molecules of the protein. And if I looked at one of your diagrams, you can clearly see where there are two separate molecules. One's oriented one way and one's the other way. And they look like they're separate. So does the computer program help you to analyze and isolate this is one molecule of the protein? Or is that something you do visually, or how does that work?
So sometimes a molecule will form a dimer. Like that particular protein, it was a dimer. Sometimes they're trimers. Sometimes it's a monomer. It all depends upon the structure of the crystal and also what's in the unit cell, which is what is that one unit that makes up the multiple repeating segments that you have in the crystal, right? Go ahead, Kelly.
Well, so, and also you have to keep in mind that every protein, the primary structure is a series of amino acids. So that's really how you determine where one molecule starts and another one ends, is you have this, this string of what they call residues.
Right. And
so
so
fed into the computer.
Right. So to get back to Judy's question, so when you see the two molecules there, those are actually two monomers. That particular protein works as a dimer. So in that particular case, we were actually able to capture it as the dimer. And the reason we did that is because there was a ligand in there. So when there isn't a ligand, that's the TRMD, the tRNA methyltransferase. So the ones without a ligand, you could see that they were just the individual monomer ones. But they usually that protein functions as a dimer. And so they were able to capture it as the dimer. But if you look in the PDB, there are different structures of it where some of them are dimers and some of them are monomers. And it's just how it was captured. Yep.
And before we get to, if anybody else has any other questions, that's what we're doing tomorrow. Tomorrow, I'm going to walk them through the PDB where all these proteins are and where you can find out what has been resolved and what related proteins there might be and such. So students, it's not like if you're joining on your phone, it's not absolutely necessary that you have access to a laptop or an actual like computer tomorrow, but it might help a little bit. It might be harder to navigate around the PDB on your phone.
Right. Because the PDB is where we get all of our information from, for not just the proteins, but the experimental data and all that. But what we all just did today was, this is really the focus of why we use Brookhaven National Lab, because everything else that we did before this can be done in a classroom, but the one thing you can't do in a classroom is use X-rays to analyze the proteins. And so what Spark does is it gives us access to multiple beam lines that we can use to specifically pinpoint the molecular structure of a protein. So it's pretty wild. And these beam lines are massive. When we go there, you'll see how big everything is. And there's one beam line that we've worked at, which is the NYX beam line. The cost of that beam line was approximately 80 million, yes, with an M, $80 million just for the beam line. And they're constantly upgrading it. They're putting new robots in. I think the robot, Kevin had said, was like $300,000 or $400,000 just for the robot. And the detectors themselves, those are extremely expensive. Those can run anywhere from $400,000 to $600,000 depending upon what you want. And actually, he just got a new detector recently too. I remember he had a problem with his detector. He had a problem with the robot. They called it the Terminator because it kept freezing on them. So it's amazing how much these beam lines cost and just how much they're constantly upgrading them so that they can keep everything working. But it's all with the intention of being able to pinpoint protein structures and identify ways that we can, basically modify these proteins and treat diseases. That's really what the focus of this is to help humans and advance humanity, right? So that's it for today's presentation. We only had one scheduled for today. So again, nice day out there. There is a little haze. I don't know if you guys noticed. There's some smoke coming in from some of the wildfires in Canada, but it's up in the upper atmosphere, so it shouldn't be making its way down to the ground like it did last year. But you may notice, like, oh, it's a little hazy out. What's with that? That's actually the wildfire smoke that's up there. So a little science lesson there for today, in addition to what we're doing. So that's it. I see you shaking your head, Kelly, like not again. No, I know. You can't get it. It's either we have hazy, hot, and humid, and we can't breathe, or we have smoke in the sky. So, yeah, it's because of the northwest wind flow. That's why. Students, any questions? Anybody have any questions about the rest of the week or next week, what we've done today or the presentations? We're good? All right. Get out, enjoy the summer, and we'll see you guys tomorrow, same time, same place. Have a great day, guys.
Bye
Have
all.
a good day, everybody. Bye.